Imagine a tiny genetic glitch that could be the Achilles heel of some of the deadliest cancers. Australian scientists have made a groundbreaking discovery that could revolutionize the treatment of highly aggressive and difficult-to-treat cancers by targeting a lesser-known genetic process called minor splicing. In a significant breakthrough reported in EMBO reports, researchers from the Walter and Eliza Hall Institute, WEHI, have found that blocking minor splicing, a rare RNA processing event, can drastically slow down the growth of tumors in liver, lung, and stomach cancers. What's even more remarkable is that this approach seems to leave healthy cells largely unharmed. Cells use a process called splicing to convert long strands of RNA into messenger RNA, which is essential for building proteins. While the majority of this work, about 99.5%, is managed by the major splicing system, a small but crucial 0.05% of genes depend on the minor splicing system. Despite affecting only around 700 out of 20,000 human genes, minor splicing plays a vital role in regulating cell growth and division. When minor splicing is inhibited, cancer cells accumulate DNA damage, which triggers a natural tumor-suppressing response that leads to cell death. This is the part most people miss. Healthy cells, however, remain largely unaffected by this disruption, making this a promising and potentially safer treatment option. Many genes regulated by minor splicing are often hijacked by cancers driven by KRAS mutations, which are among the most common genetic alterations in solid tumors. KRAS mutations are notoriously difficult to treat, and despite decades of research, progress has been limited. But here's where it gets controversial. Instead of targeting specific mutations, which might only benefit a subset of patients, the WEHI team is disrupting a fundamental cellular process that these fast-growing cancers rely on. Professor Joan Heath, head of the WEHI laboratory, explained, KRAS mutations come in various forms, making them extremely challenging to treat. Our approach is different. We're targeting a universal process that these cancers depend on, which could potentially help a broader range of patients. Using zebrafish and mouse models, as well as human lung cancer cells, the WEHI-led study demonstrated that reducing the activity of a protein encoded by the RNPC3 gene, crucial for minor splicing, significantly slowed tumor growth in liver, lung, and stomach cancers. Dr. Karen Doggett, the lead author of the study, noted, by simply halving the amount of this protein, we saw a dramatic reduction in tumor size, which is particularly impressive given the resilience of these cancers. The study also revealed that inhibiting minor splicing activates the p53 tumor suppressor pathway, a key defense mechanism in the body's fight against cancer. Known as the guardian of the genome, p53 responds to DNA damage by halting cell division, initiating DNA repair, or triggering cell death. Since p53 is often mutated or disabled in many cancers, this new approach could be especially effective in cancers with a functional p53 pathway. To identify compounds that could inhibit minor splicing, the research team screened over 270,000 drug-like molecules at the National Drug Discovery Center at WEHI. They identified several promising candidates, setting the stage for further development. This research validates minor splicing as a compelling therapeutic target, Prof. Heath said. Now, the challenge is to develop a safe and effective drug compound to inhibit it. The study's success is attributed to the diverse models and tumor types used, highlighting the potential of this strategy across multiple forms of cancer. We tested various cancer types and used multiple analysis methods, giving us confidence that our approach could be widely applicable, Prof. Heath added. What do you think? Could this new approach be the game changer in cancer treatment that we've been waiting for? Share your thoughts in the comments below.